Welcome to the DNA Extraction Lab. Our objective is to extract a visible DNA sample from strawberries. In this lesson, we'll guide you through the setup and highlight some key points to keep in mind during the lab. DNA is found in all living things. It carries genetic information and is passed from parents to offspring, providing instructions for producing the proteins that control an organism's traits. The structure of DNA resembles a twisted ladder and is described as a double helix. Since DNA is extremely small, it's usually not visible to the naked eye, or even a common microscope. But by extracting the DNA from many cells at once, we can create a mass of genetic material that's big enough to see. In today's lab, we'll use several different materials to extract the DNA from strawberry cells. Like all plants, strawberries are eukaryotic organisms. What parts of the cell must be broken apart to access the DNA from a strawberry? Strawberries are plants and they have eukaryotic cells. So, to access the DNA of this organism, we'll have to break down the cell wall, the cell membrane, and the nucleus, which is surrounded by the nuclear membrane. Let's go over the materials you'll need for this lab. Keep in mind, the materials and procedure in your classroom may be slightly different than what's shown here. In this lab, we'll be using extraction buffer, ice, isopropyl alcohol, strawberries, beakers, coffee filters or paper towels, graduated cylinders, resealable plastic bags, and toothpicks or pipettes. Remember, lab safety is key. Always follow safety guidelines and your teacher's instructions. Safety reminder: do not consume any of the materials used in this lab. Select 2 medium-sized strawberries. Make sure the leaves are removed. Place the strawberries into a plastic bag and seal it shut. Avoid sealing any extra air inside. Squish the bag for a few minutes to completely pulverize the fruit. During this step, the cell walls of the strawberries will begin to break down. Note: press firmly enough to mash the strawberries, but do not hit the bag against the table, which could cause it to burst open. When the strawberries are mostly liquidized and there are no large chunks remaining, measure 15 milliliters of the extraction buffer in a graduated cylinder. After carefully opening the bag, add the buffer to the strawberries. Reseal the bag, again pressing out any extra air, and squish for a few more minutes. Note: during this step, try to avoid making bubbles. Too many bubbles can make mixing more difficult and may interfere with the filtration process. This extraction buffer is made of salt water and soap, which helps to dissolve phospholipid bilayers. What part of the cells does this break down? The membranes surrounding the cell and the nucleus are made of phospholipid bilayers. The soap in the extraction buffer helps break down this part of the cell to access the DNA. Once the membranes are broken down, the salt in the extraction buffer helps to remove proteins that are bound to the DNA, making it easier for the DNA to clump together. When the extraction buffer is thoroughly mixed into the strawberry liquid, place a coffee filter or paper towel over a small beaker. Open the bag and carefully pour the strawberry buffer mixture through the filter. Let it drip for a few minutes as the liquid separates and collects in the beaker below. Do not squeeze the filter during this step. When about 5 to 10 milliliters of liquid has collected in the beaker, remove the filter and throw it away, along with the remaining strawberry pulp. Then, measure 10 milliliters of isopropyl alcohol into a graduated cylinder. Pour it gently down the side of the beaker, not directly into the strawberry liquid. This helps the layers of liquid remain separate. Observe the beaker for 3 to 5 minutes. What do you observe at the surface of the liquid? DNA is soluble in water, but not in alcohol. So when isopropyl alcohol is added to the beaker, the DNA precipitates, or comes out of the water and clumps together as a visible mass. Remember, during the experiment, be sure to keep a record of your data and observations. You will use this information to analyze and interpret your results. As the precipitation of the DNA completes, you can remove it from the solution with a toothpick or pipette for further observation. We can also repeat this experiment to compare the DNA from different fruits or plants, or to observe what happens to the DNA in different environmental conditions. What is one variable you'd like to test in the DNA extraction lab? There are lots of different organisms that we can easily extract DNA from. Kiwis, bananas, onions, and blueberries are all popular choices for this lab. We could also observe how the DNA responds to different environmental conditions, such as high heat. How prepared do you feel for the DNA lab? What questions do you still have? It's okay if you still have questions or feel confused. Take time to review any parts of this lab that you're unsure about. And remember, science has a lot of unknowns. Your teacher is a great resource as you begin your investigations. Good luck experimenting with DNA in the lab.